Hello, my name's Alex Wagner. I'm a postdoc at the McDonald Genome Institute at Washington University. Today I'm going to be talking about harmonization of clinical interpretations of variants. This is a project of the Variant Interpretation for Cancer Consortium, which is one of the GA4GH driver projects. A bit about the VICC. We formed actually out of the GA4GH as a working group in 2016 at the AACR GA4GH meeting. The constituent institutions participating in this consortium are from a multitude of countries, including the USA, Spain, and the UK, and include the institutions shown here, among others. Our mission is basically the global integration of knowledge bases containing clinical interpretations of cancer variants. So the idea is that a patient has a set of variants observed in a tumor, and that we can use these knowledge bases to infer what the possible clinical actionability is based off of those variants. Our ultimate goal is to provide expert-curated interpretations integrated into clinical reports. Now, the constituent knowledge bases of the consortium are shown on the left, and on the right are a number of other publicly available cancer interpretation knowledge bases. These are in addition to a multitude of ad hoc databases found at academic centers and hospitals, as well as in industry. All of these knowledge bases are effectively siloed. They all run different standards. They all have different data and curation models. And as a result, variant interpretations are highly heterogeneous. In particular, variant representation across knowledge bases is quite genetically heterogeneous. Is there a laser pointer here? Is this? Great. So as an example, there are 415 variants that are observed in, let's see, this is the third column, in molecular match. But in none of the other knowledge bases. There are 185 variants that are observed in interpretations across the OncoKB and Cancer Genome Interpreter knowledge bases, but not observed in any other knowledge base. And these colors represent basically a number of knowledge bases in the set. As a consequence, it becomes very clear that the vast majority of variants that are observed in interpretations describing clinical interpretations for variants in cancers are found in only one or two knowledge bases across this set of 6 that we evaluated. In fact, it's not until you get way out here in the tail that you begin to see variants that are observed in ubiquitously across them. And those 8 variants are shown here. And notably, less than 10% of all variants observed across these knowledge bases are found in at least half of them, and over 76% are found in only one. So this speaks to the very diverse nature of the content across these knowledge bases and the need to harmonize across them. In addition, variant interpretations are structurally disparate. So here are 3 very popular knowledge bases that are part of the consortium. So there's OncoKB, the Civic database, and the Precision Medicine knowledge base. And you'll note that the structure of the various elements that constitute a cancer variant interpretation are quite different. Those are highlighted here. And in some cases, the fields are missing entirely from a resource. It's just something the resource doesn't choose to focus on, or provides only as a free text interpretation. And so there's quite a bit of effort that needs to go into parsing and understanding and interpreting the records found in each one of these knowledge bases, a burden that before the VICC project basically meant an oncologist or other genome scientist would have to be aware of the nuances of each of these resources in order to compile a comprehensive report. Here's a cartoon representation about kind of the diversity of these elements across these 6 knowledge bases. These white pills inside each one of these larger blue pills represent interpretations, which are split up into 5 different elements: genes, variants, diseases, drugs, and evidence. And the types of data found within them can range anywhere from reference-linked data, so these are things like ensemble or entree gene identifiers, to standardized but not reference-linked materials such as commonly used disease names, to resource-specific nomenclature. So these are like evidence levels as described by each one of these knowledge bases, and they all have their own independent standards. Our goal was to take all of these elements and harmonize across them using well-known and recently emergent standards, such as the AMP-ASCO-CAP guidelines for somatic classification of variants, and provide a harmonized data store onto which we could make queries and return a partition list of results in a decreasing order of clinical significance or interest. And we did so. And I'm skipping over the details of how we did it due to time constraints. But basically what we found is that harmonization did increase breadth of interpretation. The way we evaluated this was using the AACR Project Genie dataset. So for those of you that aren't familiar with this dataset, it's a registry for cancer genomic data with clinical outcomes. And it really represents real-world data. So all of these data come from CLIA-certified labs, from major international genomic centers, and it's highly enriched in examples of late-stage disease, like many patients are when they come into the clinic. You can learn more about this project at the following link. The Project Genie cohort, we use the version 3 data release from January. It has 38,000 patients, 263 variants from those patients, and 539 distinct oncotree disease terms. You can find out more about that data set at the following URL. And what we did is we took those data and we queried all of the variants from a given patient across the 6 harmonized knowledge bases. And if there was an interpretation for any of the variants observed in a patient, we said that there was a match to that patient in that knowledge base. And then if at least one of those 6 knowledge bases contained a match, then we said in aggregate there was also a match. And after tallying that, we observed that there was somewhere around 40%, 30 to 40% on average for each one of these individual knowledge bases in identifying interpretations from this cohort. And in aggregate, we were closer to like 57, 58%. But then we extended this analysis to also query for patient disease, where we provided both the variants and the disease of the patient and searched for matches across both of those fields. And again, tallied up individually and in aggregate what the match percentage looked like. And we see reduced results, where here we've got roughly 15 to 20% across these knowledge bases individually, and closer to 30% in aggregate. And then finally, we added a tier 1 filter. So this AMP-ASCO-CAP guidelines I was talking about separates variant interpretation classifications into several tiers. Tier 1 are those interpretations that are considered of strong clinical significance. And after doing that filtering, the results are further reduced. We get down to about 10% or so on average, and close to 20% in aggregate. And what this represents, of course, is that by aggregating across these different knowledge bases, there's a lot to be learned from the patients that can show up in the clinic. And my personal impression is that with the addition of other knowledge, federated knowledge sources, that this aggregated content would only continue to climb. This represents a conservative estimate of what that interpretation matching would look like. A more liberal estimate based off of looser search criteria can go quite a bit higher. We did not provide these data for tier 1 because those standards do require very specific matching criteria in order to make the classifications valid. But in the cases of variants and variant disease matching, we noted almost a 2-fold increase in the cohort interpretability by making a broader search strategy. And there are more details about that in our associated manuscript. Now, all of this data can be found online to be, searched and explored. You can find it at our search.cancervariants.org website. This is kind of a preliminary or, prototype web interface. We're currently at the point of seeking feedback and working on our second iteration of this. All of this work is described in a preprint that we released in July, and we currently have a manuscript under review describing this as well. And I'd like to just take the final slide to acknowledge the folks over at OHSU, Brian Walsh, Jeremy Gex, and Georgia Mayfield, who were instrumental in doing a lot of the software development for this project, as well as the VICC leadership, including Obi and Malachi Griffith over here, who are my co-mentors at WashU, as well as Nuria, David, and Adam, who are also leadership, as well as our key knowledge-based partners. I also like to acknowledge the National Cancer Institute for the postdoctoral fellowship that allowed me to do this research, as well as the GA4GH for allowing us to participate as a driver project. And with that, next talk.
Yep. Okay. Great. Wonderful. So, I know it's hard work a little bit that we're going from some technical ends all the way through to clinical ends. Keep with it. Note down your questions because we'll be asking the speakers to come up here. So if you had a burning question, type it in, put it on your, scribble it down, type it into your mobile phone so you can answer the questions, ask questions.